Welcome back to the Deep Dive. Our mission today is to take on, well, one of the most critical and complex subjects in modern biology, cancer.
And we're going to do that by focusing our lens exclusively on the detailed molecular picture that's presented in the textbook, Cell and Molecular Biology.
Right, so this chapter is our entire universe for this discussion.
It is. This is a massive topic, but we're treating this textbook chapter as our sole source of truth. The goal is to distill the fundamental mechanisms, the key players, the genes, the pathways, the experimental evidence.
And deliver a precise step-by-step understanding of how a normal cell goes rogue and how scientists are trying to stop it.
Exactly.
Okay, let's unpack this right from the start.
Yeah.
What is the core definition we have to establish? The chapter identifies cancer as fundamentally a genetic disease, but we need to be careful with what that means.
That is the essential starting point. It is a disease traceable to specific alterations, mutations within the genome. However, the critical distinction, and this is really important, is that in the vast majority of cases, these are not inherited diseases.
Right. It's not usually passed down.
No. While inherited predispositions exist, of course, the genetic alterations that actually lead to the tumors arise in the DNA of a somatic cell, so a non-reproductive body cell, during the individual's lifetime.
So if you trace the cancer back far enough, you hit a corrupted piece of DNA, but that corruption wasn't necessarily there at conception. It's an acquired genetic corruption. It builds up over years.
Precisely. It's a somatic evolutionary process happening inside your own body.
Now let's paint the big picture of what happens when a cell turns malignant. What are the regulatory restraints that define a normal cell that, well, the cancer cells successfully break down?
A normal cell lives under very strict house rules. It won't divide unless it gets an external growth signal.
Okay.
It knows when to destroy itself via apoptosis if it gets significantly damaged. And it respects its spatial boundaries. It stays put.
And cancer cells?
They systematically disregard all of these homeostatic controls. They acquire uncontrolled proliferation.
And that transition from a contained, localized tumor to a deadly threat is all about the concept of invasion and metastasis.
That's the real danger. And we need to visualize this clearly, maybe by thinking about the visual evidence in figure 16.1. That image shows a malignant melanosarcoma literally invading and displacing normal liver tissue.
Just pushing everything else out of the way.
It is. And as long as the tumor is localized and hasn't breached its surrounding capsule, surgery is usually curative.
Well, once it's malignant, everything changes.
Once it's malignant, metastasis begins. Renegade cells break away. They enter the body's transportation system, the lymphatic or vascular circulation, and they travel to distant sites, establishing these secondary lethal tumors.
And at that point, the disease is system-wide. Localized treatment isn't enough.
Exactly.
Despite this daunting reality, the chapter does emphasize the massive research efforts underway, and it notes some significant progress. Figure 16.2 provides the health context, showing a tangible drop in death rates between 1990 and 2007.
Yeah, a 22% reduction for men and 14% for women. And that progress is largely attributed to earlier diagnosis of really common cancers like breast, prostate, and colon.
We're catching them when they're still localized.
Right. But this success really underscores the fundamental limitation of traditional treatments, chemotherapy and radiation.
And that limitation is the lack of specificity.
Exactly. These treatments kill rapidly dividing cells indiscriminately. That includes healthy cells in your bone marrow and your digestive tract, and that leads to these severe dose-limiting side effects.
We're constrained by how much we can hurt the patient.
Absolutely. And this necessity is the driving force behind the entire field of targeted therapies that we'll explore later. The need for precision medicine that kills only the cancer cells.
All right, let's dig into that. To develop those therapies, we first have to understand the fundamental shared characteristics of cancer cells, no matter where they start. So section 16.1, basic properties of a cancer cell.
And we start with how they're studied in the lab. They're either cultured directly from tumors, or by transforming normal cells in vitro using chemicals or radiation or even tumor viruses.
So when you look at them in a dish, what's the first thing you notice?
The defining trait is the loss of growth control. And it's not necessarily that cancer cells divide faster than normal cells, but their decision to divide has become internally driven. It's not regulated by the environment anymore.
The visual evidence in the textbook, figure 16.3, demonstrates this really well. Can you walk us through that comparison?
Sure. If you take normal cells and place them in culture, they proliferate until they cover the entire surface of the dish. And they form this neat, Single layer, a monolayer.
Just one cell thick.
Exactly. And once they contact their neighbors, their growth rate dramatically decreases. This is often called contact inhibition. They respect boundaries.
And the malignant cells.
They completely ignore these inhibitory signals. Figure 16.3 shows the resulting chaos. They just continue to grow, piling up on top of one another to form these multi-layered clumps, or what researchers call foci.
So they're just unresponsive to the cues that say, stop dividing, the surface is full.
Completely unresponsive.
This loss of control means they've achieved independence from external growth factors, which is another key concept, right? And it's illustrated by figure 16.4.
Right. Normal cells are entirely dependent on these exogenous growth factors, like epidermal growth factor or insulin, things that are typically supplied in the serum in the medium.
And if you take the serum away,
If you starve normal cells of serum, their proliferation just flatlines. But cancer cells, they continue this exponential growth even in serum-deprived conditions.
it's like they've removed the ignition key and replaced it with a permanent direct wire to the starter motor.
That's a perfect analogy. Their internal cell cycle machinery has become decoupled from the necessary external growth factor receptor interactions. This independence is a classic malignant feature.
Okay, next up is the concept of immortality. We know normal cells have a limited capacity for division before they enter, aging or senescence, but cancer cells bypass this.
Immortality is conferred by the activity of an enzyme called telomerase. The mechanism involves the protective caps on the ends of our chromosomes, the telomeres.
And they get shorter every time a cell divides.
They do. And this shortening eventually signals the cell to stop dividing. It's hypothesized to be a major defense mechanism against tumor growth in most normal cells, which typically lack telomerase.
But cancer cells flip the switch.
They activate telomerase. And by maintaining telomere length, they effectively grant themselves unlimited division potential. They become immortal, overcoming that natural time limit imposed on cellular lifespan.
Moving inside the nucleus, we hit a key molecular signature: genetic instability. Normal cells maintain a stable, diploid chromosome content, but cancer cells are chaotic.
They are profoundly unstable. And this leads to something called aneuploidy, which is just an abnormal chromosome number. It's a hallmark of malignancy.
And if we look at figure 16.5, which is a karyotype of a breast cancer cell, the visual chaos it's striking.
It is. Instead of two neat pairs of each chromosome, you see chromosomes of highly variable shapes.
Yeah.
You often see translocation, so parts of one chromosome attached to another, plus extra copies or missing copies. It is a highly deranged, fragmented genome.
What causes this internal chaos?
It results primarily from defects in the machinery that oversees cell division, so things like failures in the mitotic checkpoint or having an abnormal number of centrosomes which guide chromosome separation.
And here's the link back to a key player we'll discuss later, P53. Normal cells with this much damage would just self-destruct.
They would trigger apoptosis. But cancer cells survive this genetic chaos because they have often simultaneously disabled that apoptotic response.
So the genetic instability provides the fuel for evolution within the tumor, but the failure of apoptosis allows the damaged cell to live long enough to exploit that instability.
That is the perfect summary of the survival mechanism.
Before we move on, we have to talk about the metabolic shift, famously known as the Warburg effect, or aerobic glycolysis.
This is a really counterintuitive observation. Otto Warburg noted decades ago that many tumor cells rely heavily on glycolysis sugar breakdown for energy, even when there's plenty of oxygen available for the much more efficient process in the mitochondria.
Wait, aerobic glycolysis. So glycolysis yields far less ATP per molecule of glucose. Why would the cell choose inefficiency?
This is where the crucial insight comes in. It's about more than just energy production. While glycolysis is less efficient at generating bulk ATP, it produces ATP at a much faster rate.
Okay, speed over efficiency.
Speed over efficiency. But even more importantly, for a rapidly dividing cell, the intermediate metabolites it produced during glycolysis are diverted away from the TCA cycle and instead become precursors for crucial biomass components.
Like what?
Nucleotides, lipids, amino acids, all the things the cell needs to build two daughter cells.
So it's not about optimizing energy efficiency, it's about optimizing the speed of component production for rapid proliferation.
Exactly. They are sacrificing caloric efficiency for construction speed.
And this metabolic preference has a direct application in diagnosis, right?
It does. Since these cells are ravenously consuming glucose, we can use that in a clinical setting. This increased glucose uptake by tumor cells is tracked using PET scans, positron emission tomography.
So you give a patient radioactive sugar?
A radioactively labeled glucose analog, yes. And physicians can then locate metastatic tumors throughout the body because they literally glow on the scan due to their high glucose consumption.
And finally, related to metabolism, the cancer cell's response to low oxygen, or hypoxia.
Hypoxic conditions frequently exist deep inside a fast-growing tumor. In response, cancer cells activate a transcription factor called HIF, the hypoxia-inducible factor.
And HIF does more than just help the cell survive low oxygen.
Oh, much more. It induces new blood vessel formation angiogenesis, and it promotes migratory properties. Both of those are critical steps for the tumor to spread.
So we've defined the internal mechanisms. Now let's look outward at section 16.2, the causes. We start with history. The earliest known correlation between an external agent and cancer, attributed to Percival Pott in 1775.
Yeah, a British surgeon. Pott was farsighted enough to correlate the chronic exposure to soot with the high incidence of scrotal cancer in chimney sweeps.
And this historical observation, it really encapsulates the fundamental truth, right? The vast majority of cancer causes involve agents that damage the genome.
The mutagenic link. Whether we are talking about carcinogenic chemicals, ionizing radiation, or UV radiation, which is the leading cause of skin cancer, they all share this property of altering the genome.
They're mutagenic.
They're either directly mutagenic or they're converted into mutagenic compounds by the cell's own enzymes. The environment is constantly throwing curveballs at our DNA.
And speaking of environmental agents, tumor viruses played a critical role in the research history of cancer.
A huge role. We broadly divide them into DNA tumor viruses, like polyoma, SV40, and RNA tumor viruses, or retroviruses.
Like the one Rusals discovered.
Exactly. And the fundamental mechanism is that these viruses carry genes whose products interfere directly with the cell's normal growth-regulating activities. They essentially hijack the cell cycle.
But the chapter makes it clear that while they were indispensable as research tools, viruses are only directly associated with a small number of human cancers worldwide.
Yes, but that small percentage still amounts to millions of cases. The most crucial example is human papillomavirus, HPV. It's present in around 90% of cervical cancers and is linked to a growing number of mouth and tongue cancers.
And the good news there is the vaccine.
A huge victory in cancer prevention. The existence of effective vaccines against HPV is a game changer.
Other major players include hepatitis B, Epstein-Barr virus.
Correct. The takeaway here is that persistent viral infection can introduce foreign, growth-promoting genes into the host cell.
Now, a relatively recent and incredibly important cause that's gaining attention: chronic inflammation.
This is a major area of exploration. Chronic inflammation, whether it's triggered by a persistent bacterial infection like Helicobacter pylori in the stomach, which is linked to gastric lymphomas.
Or something like inflammatory bowel disease.
Right. It creates a microenvironment that is just ripe for mutation and proliferation. The sustained immune response releases growth factors and free radicals that promote cellular damage and division.
The idea that chronic inflammation is a promoter is supported by the preventive action of certain drugs, isn't it?
Absolutely. Think about the nonsteroidal anti-inflammatory drugs, or NSAIDs, like aspirin. Long-term use of aspirin markedly decreases the risk of colon cancer.
And the mechanism there is what?
It's thought to be the inhibition of an enzyme called cyclooxygenase 2, or KEOX2, which suppresses intestinal polyp formation. This strongly backs the theory that managing inflammation is managing cancer risk.
Let's look at the broader epidemiological picture lifestyle factors. The chapter uses migration studies to highlight the dramatic power of the environment.
Figure 16.6 is a powerful example of this. When Japanese people migrate to the US or Europe, we observe a rapid shift in their cancer incidence profile. Gastric cancer, which is common in Japan, declines.
But at the same time,
Simultaneously, their risk for colon and breast cancer, which are common in Western societies, rises sharply to match that of their new home country.
that pattern speaks volumes. Genetics might give you the susceptibility, but the environment, particularly diet and lifestyle, determines the expression.
Precisely. And within lifestyle, obesity is a major risk factor. High cancer rates among the obese are strongly correlated with elevated levels of insulin and insulin-like growth factor, IGF-1, which are themselves potent growth-promoting signals.
And the reverse is true as well. The chapter lists protective dietary components.
Yes, compounds like isoflavones in soy, sulforaphane in broccoli, and EGCG in tea are all associated with reducing risk.
And we can come back to preventive drugs too.
Definitely. Besides NSAIDs, we have tamoxifen and raloxifene, which interfere with estrogen signaling to reduce breast cancer risk. There's finasteride for prostate cancer. And even the diabetes drug metformin, which lowers cancer risk, potentially by reducing those insulin and IGF-1 levels.
Yeah, so it all reinforces the idea that what we consume and what we expose ourselves to profoundly dictates our cancer risk profile.
Absolutely.
Okay, now we enter section 16.3, the genetics, which is really the operational heart of the chapter. Let's start with the fundamental rarity. Cancer is a remarkably rare event, and it arises from the uncontrolled proliferation of a single wayward cell.
It is monoclonal. Imagine the cumulative failure. You have trillions of cells, billions divide every day, yet malignancy only results from the accumulating permanent genetic alterations in one single cell line that manages to escape all surveillance mechanisms. And it often takes decades to fully progress.
Which defines tumorigenesis as a multi-step process. It's not one mutation, it's a progression.
Each somatic mutation is like a permanent alteration to the cell's operating system. One change might grant immortality, another might offer protection from apoptosis, a third might improve invasiveness. These changes accumulate through serial selection.
Resulting in a cell line that's increasingly aggressive and unresponsive.
Exactly.
So, which cells are most likely to start this whole runaway process? Figure 16.7 looks at the cell of origin.
Right. Tumors typically arise in tissues with high cell division rates. A tissue contains stem cells, progenitor cells, and then differentiated end products.
And stem cells are the ones that are long-lived.
They're long-lived and have unlimited potential, making them prone to accumulating mutations over a lifetime. The chapter proposes that tumors can arise from either stem cells or progenitor cells, and that the cell type that initiates the tumor may determine the subtype of cancer that develops.
And once the process starts, the environment within the tumor acts as a brutal natural selector.
Absolutely. Cells within that tumor are competing fiercely for resources, and any trait that confers a growth advantage like activating telomerase, as we discussed, will be massively favored.
And the entire tumor population will eventually be composed of the descendants of the most successful, most aggressive variant.
That's natural selection in a microcosm.
And we noted earlier that not all these changes are straight mutations. That brings us to epigenetic changes.
Epigenetics refers to heritable changes in gene expression that do not involve changes in the underlying DNA sequence. Instead, they involve alterations to the chromatin structure or DNA methylation patterns.
And telomerase activation is often epigenetic.
It is. It's often an activated gene that was normally repressed.
Why are these epigenetic changes as permanent and dangerous as a mutation?
Because they're permanent and inheritable by the progeny cells. If a tumor-suppressor gene is silenced by DNA methylation, every single daughter cell inherits that silence.
And this genetic and epigenetic instability continues throughout the tumor's progression.
It does, which is why treating advanced tumors is so difficult. They're constantly evolving, constantly generating new drug-resistant populations.
We can even see this progression at the microscopic level, through histology. Figure 16.8 shows the contrast between normal and precancerous cells.
Histological changes reflect the underlying genetic chaos. Figure 16.8 contrasts healthy cervical epithelial cells, which are uniform with small, neat nuclei, with abnormal precancerous cells that are heterogeneous in shape and feature these large, messy nuclei.
And that's what a pap smear is designed to detect.
Exactly. Precancerous changes where growth control is lost even before the ability to invade or metastasize has been acquired.
What about benign tumors, like a common mole? The chapter discusses those entering senescence.
Right. Benign tumors are typically masses that pose little threat because the component cells, for example, pigment cells in a mole, have entered senescence. It's a permanent state of growth arrest.
It's just another defense mechanism.
It's another critical pathway the body uses to restrict cancer development, often triggered by early oncogenic stress.
Let's get to the core concepts then. If we use the car analogy, we have the brakes and the accelerators. The tumor suppressor genes are the brakes.
That analogy is perfect. Tumor suppressor genes encode proteins that actively restrain cell growth, monitor DNA fidelity, and promote apoptosis.
Their existence was first suggested by cell fusion studies, right?
Yes, where fusing a normal cell with a cancer cell often resulted in the malignancy being suppressed by the presence of the normal genome.
And the most important defining characteristic, which is illustrated in figure 16.9a.
They act recessively. You must eliminate or mutate both copies or alleles of the gene before the protective function is completely lost. You need 2 hits to disable the brakes.
And the flip side is the oncogenes, the accelerators.
Oncogenes encode proteins that actively promote proliferation, genetic instability, and malignancy. And they originate from normal cellular genes called proto-oncogenes.
So they're normal proteins that have been hijacked.
Exactly. Hijacked or mutated to become constitutively active.
And they are dominant. Figure 16.9b shows that.
They are dominant. A single activated copy, a single hit of an oncogene, is sufficient to cause the altered phenotype. You only need to jam one accelerator pedal down to see the effect.
So how does a normal proto-oncogene get jammed in the on position? Figure 16.10 shows the 3 main mechanisms of activation.
Path A is the simplest. A point mutation. A single base change alters the protein's structure or function, often rendering it constitutively active.
Okay.
Path B is gene duplication or amplification, where the cell makes multiple copies of the gene, leading to excessive production of the protein.
And path C
Is chromosome rearrangement, where a translocation places the proto-oncogene next to an overly active regulatory sequence, causing hyperexpression, or it creates a dangerous fusion protein.
So malignancy usually requires this dangerous combination: the loss of the brakes, the tumor-suppressors, and the jamming of the accelerator, the oncogene activation.
Exactly. As long as the brakes are functional, they provide a degree of protection against the effects of an activated accelerator. You need both systems to fail for unchecked malignancy.
All right, let's look at the titans of tumor suppression, starting with retinoblastoma, RB. This gene defined the 2-hit hypothesis from Alfred Knudsen.
Right. RB is linked to a rare childhood retinal cancer. Knudsen hypothesized that every case requires 2 hits. In the sporadic form, which is shown in figure 16.11a, a single retinal cell must acquire 2 extremely rare successive spontaneous mutations in both RB alleles. It's a very low probability event.
But the familial cases are different.
Very different. In familial cases, as shown in figure 16.11b, the individual inherits 1 non-functional allele from a parent. Every cell in their body starts with 1 strike against it.
So they only need 1 more hit.
Only 1 subsequent somatic mutation or deletion, the second hit, is needed in any retinal cell to lose all functional POB protein and initiate tumor growth. This explains the extremely high predisposition and the early onset.
And the broader impact of RB loss is huge.
Oh, yes. Loss of RB function is a common feature in sporadic adult cancers, including breast, prostate, and lung cancers. The protein product, PRB, is the central gatekeeper of commitment to division.
Let's slow down and describe that mechanism shown in figure 16.12 because it's absolutely foundational to cell cycle control.
Okay, so PRB controls the crucial G1 to S phase transition. That's the point of no return. During the waiting period, G1, the unphosphorylated PRB, acts as a transcriptional repressor.
How does it do that?
It physically binds to a critical transcription factor called E2F, effectively keeping E2F locked up and preventing it from activating the genes required for DNA replication, the S-phase genes.
So it's holding E2F hostage to prevent division.
Correct. When the cell receives sufficient external growth signals, CDK-dependent kinases become active and they hyperphosphorylate PRB.
And phosphorylation is the release signal.
Think of it as marking PRB for release. Once it's heavily phosphorylated, PRB changes its conformation and lets go of E2F. Free E2F then rushes to the nucleus, activates the transcription of all those S-phase genes, and the cell is committed to replication.
So the consequence of losing PRB?
You lose the ability to hold E2F hostage. The cell cycle is essentially running perpetually without needing external signals, leading to uncontrolled proliferation. It's also why DNA tumor viruses evolved to block PRB function. It's the quickest way to force the host cell to replicate the virus's genome.
Now let's move to the most famous tumor suppressor, P53, the product of the TP53 gene, the guardian of the genome.
TP53 is the most frequently mutated gene across the entire spectrum of human cancers. It's altered in about half of all tumors. An inherited abnormal TP53 allele causes lifelong mini syndrome, which confers an incredibly high lifetime risk of developing various cancers.
What makes P53 such a critical monitor?
It acts as a stress-activated transcription factor. When the cell detects stress like lack of nutrients, hypoxia, or most critically, DNA damage, P53 concentration spikes, and it orchestrates 3 potential responses. responses.
And figure 16.13b shows why it fails so often.
It does. The common debilitating mutations occur right in the DNA binding domain, which cripples its ability to activate its target genes.
Okay, let's detail those three actions, starting with G1 arrest, which we see in figure 16.110.
When DNA damage occurs, it stabilizes the p53 protein, dramatically increasing its half-life and its concentration. High p53 then activates the expression of the p21 gene.
And we know P21, it's a CDK inhibitor.
Exactly. It binds to the CDK cyclin complex, inhibiting its activity, thereby freezing the cell cycle in G1 to allow time for DNA repair.
And if repair fails, action 2 kicks in, apoptosis.
p53 ensures damaged cells don't survive. It promotes cell death, partly by activating the expression of pro-apoptotic genes like BAX. BAX products initiate the intrinsic apoptosis pathway.
It can also act more directly, can it?
Yes, it can bind directly to members of the BCL2 family at the mitochondrial membrane, triggering membrane permeabilization and releasing factors that cause the cell's demise.
And action 3, senescence, is a backup.
It is. If the cell can't repair the damage and can't be easily killed, p53 can push it toward permanent growth arrest or senescence, stopping it from replicating the damaged DNA.
This complex guardian role has huge clinical implications, particularly regarding drug resistance, which figure 16.15 demonstrates.
This is a devastating insight. The figure shows that cells lacking functional TP53 are highly resistant to chemotherapy and radiation.
Why?
Because those treatments primarily work by inducing massive DNA damage in cancer cells. If the cell can't activate p53, it can't be directed into apoptosis, meaning it can simply continue proliferating even under a heavy dose of chemotherapy agents like adriamycin.
So how is P53 normally regulated so it doesn't constantly drive healthy cells into arrest or death?
It's tightly controlled by a protein called MDM2. You can think of MDM2 as a molecular executioner. It binds to p53 and targets it for destruction via ubiquitination.
So P53 levels are normally kept very low.
Very low. It's rapidly made and rapidly destroyed, but when DNA damage occurs, it activates sensor kinases like ATM. ATM phosphorylates p53 at a site that prevents MDM2 from binding. This stabilizes p53 and allows its concentration to rise high enough to activate its target genes.
So that dynamic balance, P53 the lifesaver, MDM2 the destroyer, is the essence of the pathway.
And critically, if a tumor finds a way to overexpress MDM2, it can functionally silence even a wild-type healthy p53 gene. This confirms that it's the disruption of the pathway that leads to malignancy.
Let's rapidly cover the other essential tumor suppressors in Table 16.1, starting with APC, which is crucial in colorectal cancer.
APC mutation predisposes individuals to familial adenomatous polyposis, or FAP, where the colon is littered with thousands of polyps. APC's normal function is to suppress the want signaling pathway, which promotes proliferation.
So its loss lets want signaling run rampant.
Exactly. And APC may also play a role in chromosome segregation, meaning its loss can also contribute to aneuploidy.
And BRCA1 and BRCA2, famous for hereditary breast and ovarian cancer risk.
These are not cell cycle regulators in the same way as PRB. They are DNA damage response proteins. They are vital components of complexes that activate DNA repair mechanisms, specifically homologous recombination, to fix double-strand breaks.
And as figure 16.16 illustrates, if that BRCA pathway fails,
then p53 is activated as a backup, leading to cell arrest or death. When BRCA is lost, the cell has to rely on more error-prone repair systems, which leads to genetic instability.
Finally, PTNN, linked to cell survival pathways.
PTNN is a brake on the survival signaling network. It's a lipid phosphatase that dephosphorylates PIP3, effectively turning off the PI3-KP-CASI, or AKT, cell survival pathway.
So loss of PTN is very common in advanced cancers.
Extremely, because it leads to excessive PIP3 and an overactive PKB, which promotes cell survival and inhibits apoptosis. And remember, these suppressors are often inactivated not by mutation, but by epigenetic silencing, like DNA methylation in their promoter region.
Okay, now for the accelerators. Figure 16.17 provides a taxonomy of oncogene proteins, highlighting their diverse roles, from growth factors to transcription factors to metabolic enzymes. But they all share the common goal of disrupting normal signaling.
The GTP-binding protein RAS is the poster child here. It's the most frequently mutated oncogene in human tumors. An oncogenic RAS mutant is typically stuck permanently in the active GTP-bound form,
Sending nonstop divide signals.
continuous, non-stop proliferation signals to the cell nucleus regardless of external conditions.
Let's detail the examples involving growth factors and their receptors.
We see two common methods here. One is autocrine signaling. The cis-oncogene, derived from the platelet-derived growth factor, or PDGF gene, causes cells to secrete large amounts of PDGF, stimulating themselves to divide uncontrollably.
And that's implicated in brain tumors.
Gliomas, yes. The second method involves the receptors themselves. The ERB oncogene encodes a truncated EGF receptor that is missing its external ligand binding domain.
Meaning it doesn't need the growth factor to turn on.
Exactly. It's constitutively stimulated, always sending the divide signal, even in the absence of external growth factor. Human cancers often achieve the same effect simply by overexpressing receptors like EGFR or HER2, making the cell hypersensitive to minuscule amounts of growth factor.
What about cytoplasmic protein kinases? RAF is crucial, heading the MAP kinase cascade.
RAF is central to the primary growth-controlling pathway in the cell. Mutations that leave the RAF protein permanently on convert it to a powerful oncogene. We see this clinically in BCR-ABL mutations, which are responsible for about 70% of malignant melanomas.
And MYC, the transcription factor.
MYC is a master regulator. It controls the expression of hundreds of genes involved in growth and proliferation. It's often amplified or involved in chromosomal rearrangements. The classic example is Burkitt's lymphoma, where a translocation moves the MYC gene next to a highly active antibody gene promoter,
Leading to explosive, uncontrolled overexpression.
Exactly.
This chapter also pulls in some surprising, more recently discovered oncogene types, like those affecting the epigenetic state and metabolic enzymes.
The epigenetic link is crucial because mutations in enzymes like DNA methyltransferases or DNMT3A or histone modifiers can promote tumorigenesis by improperly silencing tumor suppressors or activating proto-oncogenes. It's a layer of chaos above the DNA sequence itself.
And the metabolic enzymes, that was a surprise.
A huge surprise. Recurrent mutations were found in isocitrate dehydrogenase, IDH1 or IDH2, a core enzyme in the TCA cycle.
But the mutant enzyme doesn't just stop working.
No, it no longer performs its normal function. Instead, it produces a metabolite called 2-hydroxyglutarate, or 2-HG. This molecule accumulates and acts as a potent inhibitor of histone demethylases and other key epigenetic regulators.
So the metabolic disruption leads directly to global epigenetic chaos, promoting the tumor.
Precisely. It's a perfect example of how malignancy can result from a cascade where a mutation in one area, metabolism, cripples another fundamental control system, epigenetics.
Finally, we revisit apoptosis, looking at the anti-apoptotic oncogene, BCL2.
BCL2 is a protein that actively inhibits programmed cell death. Overexpression, often caused by chromosomal translocation, for example, in follicular B-cell lymphomas, allows abnormal cells to survive and proliferate when they should have died.
And that also contributes to chemotherapy resistance.
A major contributor, yes.
Figure 16.18 provides the necessary synthesis here, showing the entire interconnected signaling map.
It drives home the realization that cancer is not defined by one single mutated gene, but by the systemic failure of an interconnected network. You can visually trace the flow RAS feeds into RAF, EGFR feeds into the pathway, and the regulatory loops between P53 and MDM2 are all linked. Malignancy requires disrupting multiple points in these core networks.
And that leads to the concept of the mutator phenotype, driven by defects in DNA repair proteins.
Right. If you have defects in DNA repair, as seen in patients with hereditary non-polyposis colorectal cancer, or HNPCC, you acquire an abnormally high self-accelerating mutation rate. This dramatically increases the chances of incurring secondary, tertiary, and quaternary mutations in tumor suppressors and oncogenes, rapidly accelerating tumorigenesis.
We also have to mention microarrays, or miRNAs, a new tiny regulatory player.
These small RNAs regulate gene expression by targeting and negatively regulating messenger RNAs. Some act as tumor suppressors. For example, miR-15A and miR-16 normally inhibit the anti-apoptotic protein BCL2.
If you lose them.
If these miRNAs are lost, BCL2 is overexpressed, promoting leukemia. Others act as oncogenes. Their overexpression can silence tumor suppressor mRNAs, like those targeting TP53.
This means miRNA expression profiles could be critical biomarkers.
Absolutely, for both diagnosis and therapeutics. The chapter mentions the potential for miRNA sponges, which are synthetic RNAs designed to bind and sequester oncogenic miRNAs, effectively taking the foot off the accelerator.
Now for the overwhelming scale, the cancer genome. Sequencing efforts have identified over 350 cancer genes, and figure 16.19 visualizes the mutational landscape, which is incredibly complex.
When you sequence a tumor, you find a chaotic landscape. There are mountains. These are high-frequency common mutations like APC, KRAS, and TP53, which are the assumed drivers of the disease. But then there are hundreds of low-frequency hills, which could be secondary drivers or just harmless passenger mutations.
And the crucial takeaway from comparing two different tumor sequences, even of the same type, is how few mutations they often share.
That heterogeneity is the core challenge. It suggests that each patient's tumor is a genetically distinct disease.
Yet, despite this individual variation, Figure 16.20 shows that the progression often follows a sequence.
It does. In colorectal cancer, APC loss is an early event, followed by KRAS mutation, then later TP53 mutation. Chromosomal instability dramatically increases late in the progression.
And this leads us to the most vital modern insight, summarized in Table 16.2. Cancer is a disease of pathways, not just individual genes.
This is the hope. While the specific gene that is mutated might differ wildly between patients, those mutations almost always converge on disrupting a small core set of cellular pathways: apoptosis, the G1S transition, once signaling.
And that's where the consistency lies.
Yes. If we look at pancreatic cancer, Table 16.2 indicates that 100% of the tumors showed disruption in these key pathways. That uniformity in pathway disruption provides a much more rational basis for drug targeting than chasing individual mutations.
Before we transition to therapies, we need to touch on gene expression analysis, specifically DNA microarrays, which have revolutionized prognosis and diagnosis.
DNA microarrays allow us to look at the activity profiles of thousands of genes simultaneously. Fluorescently labeled cDNA synthesized from a tumor's mRNA is hybridized to a slide containing DNA spots.
And Figure 16.21 shows its diagnostic power.
It does, clearly distinguishing between acute lymphoblastic leukemia, AL, and acute myeloid leukemia, AML, based purely on their distinct patterns of gene activity.
And its prognostic value is truly life-changing, as shown with the breast cancer data in Figure 16.22.
This is the real-world application. The gene expression profile allows clinicians to categorize early-stage breast cancer patients into good prognosis or poor prognosis groups.
So you can tailor the treatment.
Precisely. If a patient with a small, apparently contained tumor shows a poor prognosis gene signature, they are aggressively treated with chemotherapy to prevent recurrence. Conversely, a patient with a similar-looking tumor but a good prognosis signature can be spared the debilitating effects of chemo.
It moves treatment from general rules to personalized molecular guidance.
Exactly.
The knowledge of these aberrant pathways is the engine driving Section 16.4. New strategies for combating cancer. Given that conventional treatments fail with metastatic disease, we need targeted therapies that achieve specificity.
Targeted therapies aim for three things: killing only cancer cells, targeting proteins essential for the cancer cell's survival, and tailoring the treatment to the patient's specific mutational profile. We can group these strategies into immunotherapy, protein inhibition, and angiogenesis inhibition.
Let's start with immunotherapy, which has exploded recently. But it traces its inspiration back to William Coley in the late 1800s, who observed spontaneous remissions linked to immune responses.
It does, and we have passive and active approaches. Passive immunotherapy is about deploying tools directly into the patient, specifically therapeutic antibodies.
How do those work?
They're designed to recognize and bind specific proteins on the surface of tumor cells. Once bound, they can inhibit the protein's function or flag the cell for immune destruction. And advances in producing humanized antibodies have minimized adverse reactions.
What are the best examples?
Herceptin is a major success. It targets the HER2 receptor, which is overexpressed in about 25% of breast cancers. By blocking HER2 activation, it has dramatically improved survival for those patients.
And Rituxan.
Rituxan targets the CD20 protein on malignant B cells in non-Hodgkin's lymphoma, inducing apoptosis. And we can increase the lethality by using conjugated antibodies like Zevalin, which link the antibody to a radioactive atom, delivering the radiation payload precisely to the cancer cell.
Active or adoptive immunotherapy is much more complex. It aims to stimulate or engineer the patient's own immune system to overcome immune tolerance.
This is truly personalized medicine. DCVax, for glioblastoma, extracts the patient's dendritic cells, stimulates them in vitro with tumor antigens, essentially teaching the immune system what to look for, and re-injects them as a cancer vaccine. It's shown extended survival times in trials.
And the incredible high-risk work with genetically engineered T cells for chronic lymphocytic leukemia, or CLL.
This is revolutionary. T cells are isolated from the patient and then genetically engineered, often using a disabled HIV vector, to express a specific high-affinity T cell receptor, a TCR.
And this TCR is programmed to recognize a tumor antigen.
Right, like CD19 found on malignant B cells. Reinfusion of these supercharged T cells has led to complete remission in some phase I trials. The risk is high because the engineered T cells will also attack normal B cells, causing severe autoimmune side effects.
Okay, moving to the second strategy, inhibiting cancer-promoting proteins. This relies on the core concept of oncogene addiction.
Right. If a tumor has become so reliant on a single aberrant protein to sustain its growth, blocking that protein should cause the cancer cell's death. The gold standard success story here is Gleevec.
Gleevec treats chronic myelogenous leukemia, CML, which is caused by the famous BCR-ABL fusion gene,
which expresses a constitutively active ABL tyrosine kinase. Gleevec selectively binds to the inactive form of the ABL kinase, preventing its activation. Initial trials were stunning. Gleevec caused remission in almost every CML patient.
It proved the whole concept of targeted therapy.
It did. It proved that eliminating a single oncogene product can effectively stop a human cancer.
But the challenge, as always, is resistance.
Resistance emerges quickly, often through secondary mutations in the ABL gene itself, preventing Gleevec from binding. Patients need continuous treatment and often require newer second-generation inhibitors or combination therapies to stay ahead of the evolutionary curve of the cancer cells.
Another key success story is Zelboraf for metastatic melanoma.
Zelboraf targets the specific BRAF-V600E mutant enzyme found in 70% of melanomas. Figure 16.23 visually captures the initial miracle. Metabolically active tumor lesions tracked by AT scans simply disappear after a few weeks of treatment.
But again, resistance develops rapidly.
Typically within 7 months. And often because the tumor finds ways to activate downstream signaling pathways to bypass the blocked BRAF. This confirms that combination therapy targeting multiple nodes in the pathway is the necessary future.
This necessity of knowing the specific target leads to the issue of tumor heterogeneity and companion diagnostics.
We cannot treat all lung cancers or all melanomas the same way. The EGFR inhibitor Aresa, for example, failed initially in trials until researchers realized it only worked if the patients had wild-type KRAS.
If KRAS was already mutated, Aresa couldn't help.
Exactly. And the drug Elcori, an ALK kinase inhibitor, is only effective in the small subgroup expressing the EML4-ALK fusion protein. Therefore, targeted therapy demands companion diagnostics genetic testing to match the drug to the patient's unique genetic profile.
A very clever general strategy emerging from this pathway knowledge is synthetic lethality, using drugs like PRP1 inhibitors.
Synthetic lethality is a sophisticated concept. We exploit a weakness created by the loss of a tumor suppressor. Remember, BRCA1 and BRCA2 are involved in DNA repair. Cells lacking functional BRCA rely heavily on a backup repair system, PRP1, to survive.
So if you inhibit PRP1,
you strip the cell of both repair mechanisms, causing it to die selectively. Normal cells, which still have functional BRCA, can survive the PRP1 inhibition. This offers a general strategy for targeting cells that have lost tumor suppressors.
This entire discussion also forces us to confront the biological concepts of the cancer stem cell.
Right. The hypothesis here challenges the idea that a tumor is a homogeneous mass. It posits that only a tiny, highly aggressive subpopulation, the cancer stem cells, possesses the long-term potential to sustain the primary tumor or initiate metastasis.
The implication for Gleevec, or any drug, is frightening.
If Gleevec kills the bulk of the tumor mass but spares these few cancer stem cells, the tumor will inevitably recur, even after initial remission. So, new drugs must be developed that specifically target the initiating and self-renewing capacity of these stem cells.
The final strategy covered in the chapter is inhibiting angiogenesis, blocking new blood vessel formation. Figure 16.24 visually explains why this matters.
Tumors cannot grow beyond 1 to 2 millimeters without a blood supply. They achieve this by secreting angiogenic growth factors, like VEGF. This process recruits and stimulates the proliferation of endothelial cells, leading to vascularization and unlimited growth potential.
And the logic of anti-angiogenic strategies is compelling.
It is. Since we are targeting normal, genetically stable endothelial cells that form the blood vessels, rather than the genetically unstable cancer cells, we theoretically reduce the likelihood of drug resistance emerging.
So how has that worked out?
Avastin, an antibody directed against VEGF, achieved modest success. It prolonged life for months in metastatic colorectal cancer when combined with chemotherapy. But anti-angiogenic strategies have not been the panacea some had hoped for, and research interest has currently waned a bit.
Ultimately, the best strategy, regardless of molecular advances, remains the earliest possible detection.
Absolutely. The progression from an initial epithelial cancer to a metastatic disease takes at least a decade. Screening programs, mammography, pap smears, colonoscopy are vital lifesavers. The future hope is finding new biomarkers, whether mutant DNA, mRNAs, or abnormal proteins, detectable non-invasively in the blood or bodily fluids.
To properly conclude our deep dive, we have to look at the experimental pathways feature. It's dedicated to the history of these discoveries. And this is crucial for understanding how we jumped from a viral cancer in a chicken to a specific point mutation in the RAS gene.
This narrative begins in 1911 with Peyton Rouse and his groundbreaking work on a chicken sarcoma. Rouse did an incredibly elegant experiment. He took the tumor, ground it up, filtered the supernatant through filters so fine that they would block bacteria, and then injected this cell-free filtrate into healthy hens.
And the result was revolutionary. The hens developed the tumor.
It demonstrated that cancer could be transmitted by a filterable virus, an RNA tumor virus, later named Rouse sarcoma virus, or RSV. This was the birth of the viral link to cancer.
Now we fast forward to 1970, and the theoretical conflict known as the central dogma, which stated that information flows from DNA to RNA to protein. But RNA viruses needed a DNA intermediate, a provirus, to replicate.
Which implied the existence of an enzyme that could violate the dogma by synthesizing DNA from an RNA template. Independently, David Baltimore and Howard Temin discovered this enzyme, RNA-dependent DNA polymerase, or reverse transcriptase.
How did Baltimore's team prove this activity definitively?
They incubated purified viral particles, or virions, with labeled DNA precursors. They showed that the labeled DNA precursor was incorporated into a DNA product.
And the crucial control experiment.
That's shown in Figure 2. If they pretreated the virions with RNase, an enzyme that destroys RNA, the incorporation of the labeled precursor dropped dramatically. This proved that the viral RNA was acting as the template for the synthesis of the DNA copy.
This confirmed the provirus hypothesis and overturned a fundamental belief about biological information flow. But the question remained, what specific gene in that virus was causing the transformation?
That brings us to Varmus and Bishop in the mid-1970s. They used deletion mutants of the avian sarcoma virus, ASV, that could grow but couldn't cause sarcomas. They zeroed in on the SARC gene. They synthesized a labeled complementary DNA, cDNA's arc, corresponding to this gene.
And their finding when they used that probe?
It was the defining moment. They found that cDNA's arc hybridized not just to the DNA of infected chicken cells, but also to the DNA of normal uninfected chicken cells, and, astonishingly, to the DNA of every vertebrate species tested.
So the conclusion was massive. The transforming gene, VSRC, the viral oncogene, was not originally viral at all.
It was a cellular gene, a proto-oncogene named CSRC, that the virus had picked up and modified. This proved that our own cells harbor the latent potential for cancer.
What was the function of this SRC product, PP60RC?
Using antibodies, they identified PP60RC as a protein of 60,000 Daltons and confirmed its function as a protein kinase. The huge surprise was its substrate specificity.
It wasn't like other kinases.
No. Unlike all kinases known at the time that phosphorylated serine or 309 residues, PP60RC phosphorylated tyrosine residues.
And Figure 3 shows the localization of this novel kinase.
Yes, electron microscopy localized PP60RC to the inner surface of the plasma membrane. The key difference between the viral version and the cellular proto-oncogene was one of activity. The viral VSRC functioned at a much higher, unregulated level, leading to massive phosphotyrosine signaling.
The final step in this historical path involved Robert Weinberg and the realization that oncogenes could be activated purely by chemical carcinogens without a viral intermediary.
In the late 1970s, Weinberg used DNA transfection. He took DNA from chemically transformed mouse cells and introduced it into non-malignant NIH3T3 mouse fibroblasts. The formation of malignant cell clumps proved that chemical carcinogens could create stable, inheritable nucleotide sequence alterations sufficient to activate cellular proto-oncogenes.
And the link to human cancer followed immediately.
Weinberg's team showed that DNA isolated from human bladder carcinoma cells could also transform NIH3T3 cells. The transforming gene was identified as RAS, the same oncogene carried by the Harvey sarcoma virus. This confirmed RAS was key to both viral-induced and chemically-induced human cancers.
And the ultimate breakthrough was the mechanism of RAS activation.
Nucleotide sequencing revealed that the activation of RAS was achieved by the simplest mechanism. A single base substitution, a guanine, changed to a thymidine. This single-point mutation resulted in valine replacing glycine at position 12.
And that one amino acid change was enough.
An amino acid substitution sufficient to lock the protein in the act of conformation. It was a dramatic confirmation that cancer starts with the simplest of genetic errors.
What a journey. Our deep dive confirms that cancer is a disease defined by accumulated heritable defects in core signaling pathways. It's the failure of the brakes tumor suppressors like P53 and PRB combined with the hyperactivation of the accelerators proto-oncogenes like RAS and MYC.
And the path forward is clearly defined by the genetic chaos we've mapped. Treatment is irrevocably shifting away from indiscriminate cellular destruction toward highly specific, tailored pathway disruption, powered by immunotherapies and bespoke protein inhibitors like Gleevec, guided by gene expression and companion diagnostics.
That brings us back to our final provocative thought. Given the sheer scale of genetic complexity and the profound heterogeneity of individual tumors, where even two patients with the same cancer subtype might rely on completely different sets of mutations for survival, we have to ask, can human ingenuity ever devise a single universal cure? Or will cancer treatment forever remain a customized, multi-drug cocktail strategy, perpetually chasing the unique, evolving array of aberrant pathways specific to each patient's mutational landscape?
That quest for specificity is the defining challenge of modern medicine.
Thank you for joining us for this incredibly detailed deep dive into the world of cancer cell biology. We hope you feel thoroughly informed.
We'll see you next time on The Deep Dive.